missing just because large spot sizes were used. And on top of that, the entire acquisition took several hours, even for very small areas. Now, as the aerosol transfer technologies and the ablation cell evolved over time, the laser started being more efficient and even faster at delivering the aerosol from the ablation site into the plasma. So that led to an increase in the image quality and also the image resolution. So basically, that's what we're showing here on the left-hand side. So in the beginning, you had to use large spot sizes, and then the image was rather pixelated. And then as the technology got better and better, the image quality has increased as well. So now once we've managed to achieve really fast single pulse responses in a millisecond range, so to speak, the next step was to prove that the instruments were capable of handling such speed. In other words, the other aspect that Damon mentioned, the repetition rate of the lasers was put to a test. Now, together with our collaborators at the National Institute of Chemistry of Slovenia, excuse me, in Ljubljana, we looked at running really fast imaging experiments using a new generation of 500 Hz laser, and really to see what the analytical benefits of using those lasers are. And that's what we're showing here on the right-hand side. So we selected Murano glass beads, so one of those tiny and highly decorative glass objects manufactured in Venice, in Italy. And we split those, or we split that particular sample into 3 equally sized areas. And each of those areas was then ablated and analyzed using a different repetition rate. Now, if you look at the images that were generated, you will see that the quality is exactly the same. So the signal to noise ratio, the contrast is exactly the same, irrespective of the repetition rate that was used. However, the biggest difference is when you're looking at the acquisition time. So firing at relatively low rep rates, 100 Hz, the area was ablated, or the acquisition time was around 40 minutes, 45 minutes, whereas when you go up to 500 Hz, that acquisition time decreases quite a bit. So it's saving valuable time and also resources, because you don't have to keep the plasma on for so long, and you have to consume all those gases for that long. And there's a paper currently in press showing that you can go up to 1 kilohertz and still get really good data, really good quality images, and so on. Now, I think Damon mentioned earlier that initially the laser ablation ICP-MS elemental imaging was developed by geochemists. I'm not just saying that because I myself am a geochemist, but laser ablation in general, or when it started, was designed as a tool to help the analysis of minerals and so on in earth sciences. Now, as the technique gained traction, excuse me, people have started looking into the distribution of elements within samples, and have soon figured out that putting all the data into an image makes more sense. In fact, what I'm showing on the right-hand side here, so this table contains exactly the same data as it's shown in the image on the left-hand side. But because the human eye is rather trained to identify patterns more easily, the image is much easier to digest and has more impact to the viewer. And by the way, the image on the left-hand side is of a mineral called zircon, and it shows how the element thorium is distributed within the mineral, and also how lead has seeped into the grain via cracks and so on and so forth. Now, zircon grows continuously over a period of time, and its chemistry reflects the changes in the environment in which it grows. That's why you have those nice zonations, different shades of red, which reflect the changes in chemistry. It's very similar to how the tree rings capture the environmental changes that take place throughout the year in the environment. An important thing to keep in mind, and that's shown here on the lower right-hand side with the looping video, is that those zonations, or those chemical variations, are a 3D feature. And this can be also captured by using laser ablation ICP-MS. And that's what the video is showing. Basically, it's showing the distribution of uranium in a zircon grain as a function of depth as well. Now, from the analysis of mineral samples, it didn't take long for labs to figure out that the same technique can be really successfully be applied to biological samples. And more importantly, users have realized that with the same instruments, so with the new technology available, they can adapt the acquisition parameters to the needs of the applications. So going from subcellular scale basically to fully grown organs can easily be achieved. And that's what we're showing on this slide. So the image on the left-hand side shows the distribution of certain elements, phosphorus, within a cell. And because of the resolution we use, so for that particular experiment, the resolution is 0.5 microns. You can really well see the internal structure of the cell, and I'm pretty sure that image tells you more than it tells me, just because of my background. But the fact is, you can still see, or you can see the structure of the cell by using this technique. And on the right-hand side, I think this image was shown before during today's presentation. You can see a large area which was mapped at a higher or low resolution, rather 12, 15 microns, but you can still see really nice isolation, really nice distribution of the elements within the sample that was analyzed. And what I'm showing on this slide is not, the proverbial cherry-picked data. This is data that was independently generated by our collaborators in the academia, and most of the data, or all of the data rather, was published in peer-reviewed journal. And I think this is a quite important point to make, that all the hardware development and the software development that was done by photon machines throughout the years was not carried out in a bubble, so to speak, by a bunch of engineers. Everything that was designed, was developed, and was tested, it was done via collaborations with the academia. And that's a key aspect of this entire laser ablation philosophy, if you want, when it comes to photon machines. Now, we've only spoken thus far about hardware, but keep in mind that with the new technologies, you're generating huge amounts of data, and that data needs to be handled somehow. And that's how, or that's the reason, well, one of the reasons why we decided to start developing our own software packages, basically, just because that's an integral part of the equation. Good quality, and I know you already know that good quality, relevant data can be difficult to achieve, and especially for laser ablation ICP-MS, just because back in the old days, it was a difficult technique to handle and to master. We've developed a software which is called HTIP to simplify the entire process. The software was initially developed by the group at the Ghent University, in Frank van Haken's group, but then was carried over to Teledyne and developed even further. And it's now a true mass spectrometry toolbox, data analysis, and so on and so forth. And the idea behind the software is to enable even the most novice of users to make the most out of their laser ablation ICP-MS setup, basically. Now, what HTIP does and excels at is simplifying the workflow. And that's done by automating a lot of the procedures, from the acquisition parameters that you need to optimize to get really good quality data and really good quality images, to the image generation itself, and then to further data analysis. And one key aspect of HTIP is that it's continuously evolving. And the aim is to become sort of a bridge between the operating software of the laser and of the ICP-MS, irrespective of the manufacturer. So basically to offer an integrated solution for the users. Now, HJP is a pretty complex piece of software, and it's not the time or the place to do a proper software demo. And in all honesty, it will probably take the remainder of the day just to walk you through the basic features of the software. We'll be happy to arrange any demos afterwards, or if you have any questions, I'd be happy to address those afterwards. But I think it's important to recognize that the software allows the user to do a number of things and to optimize a number of procedures, from optimizing the parameters that the user needs to use to get the really good quality images, really good quality data, both on the laser ablation and on the ICP side. And that's an important thing to mention and to keep in mind, that HJP allows you to model the acquisition parameters on both instruments, both on the ICP and on the laser side. And it can do a lot of other things. It can, in some instances, it can stream data live from the instruments. It can do all sorts of image segmentation, and I know this is a key aspect for biological imaging, just because there's a lot of different techniques used within the field of imaging, and being able within one software to overimpose, image from a laser ablation experiment with a microscopy, confocal microscopy, or any other sort of image generating technique, and then perform all sorts of image segmentation procedures is quite important. Also, HJP allows the user to generate 3D images if your experiments were designed accordingly. There's a lot of things HJP can do, and in fact, a lot of the images that were shown today during the presentations, excuse me, were generated using HJP. So you had the chance to see some of the images and the quality of those images, and the output basically from the laser ablation ICP-MS technique. And with that, I think we should hand it over to our colleagues at Thermo. But as Tia mentioned, towards the end of the presentation, we'll have a live Q&A, and I'll be happy to reply to any questions you might have.
Thank you, Cyprian. Great presentation. Let me see if I can maintain the level. Okay, let's go ahead. Also, a welcome from my side. My name is Daniel Kutscher. I am a product marketing specialist at Thermo Fisher Scientific. I'm an analytical chemist by training, and I do have some metabolomics background. So this is also based on some personal experience from myself. I graduated from the University of Oviedo about 10 years ago, the research group of Alfredo Sanz Medel, which I think was, back in the days and still continues to be one of the key centers for establishing metabolomics as a scientific branch. So right now, I would like to first start to talk a little bit about the Thermo Fisher Scientific as a company, and also more specifically, the Center of Excellence for Mass Spectrometry located in Bremen, Germany. So basically slightly a little bit more to the east from London. You can see here an image of the site where I'm working at. This is quite a big site located directly at the Bremen airport, with also a large tradition in the development and manufacturing of mass spectrometers. So basically the origin of this company is dating back more than 70 years now. The first commercial instrument was brought to the market as early as 1950. And right now you can see our building here on the bottom of the slide. There's more than 450 people with more than 30 nationalities, so quite a diverse and international team working on the development of a range of different mass spectrometers, not only ICP-MS, but also other instruments, for example, like multi-collector ICP-MS, gas isotope ratio, and also the Orbitrap technology is key of the portfolio of the Bremen site. So let's talk about the ICP-MS side of things as, let's say, the second part to construct an imaging tool set that can be applied in order to determine elements in a spatial resolution across, for example, a thin section of tissue. For elemental analysis, there's a number of different instruments available, which you can all see here in this slide. And this starts, for example, with ICP optical emission spectroscopy, a technique that you can see on the left-hand side. You might recall this maybe for those of you that did analytical chemistry at university. You might have used one of these instruments back in the days. Single and triple quad ICP-MS, but there's also high resolution ICP-MS systems available with a magnetic sector, double focusing setup, and also a product recently brought to market, a new multi-collector ICP-MS with the NEOMA system. Now, similar as to what Cyprian and Damon did, I would also like to talk a little bit about the evolution of mass spectrometers of over the last couple of years. And also here, likewise, we have seen a development, for example, in the ability of higher repetition rates in conjunction with smaller spot size. Also here, we have seen a gradual response on the ICP-MS side to go for more sensitivity on the instruments in order to also be able to detect the element of interest in this small quantity that is actually ablated and brought to the plasma if you do laser ablation with, let's say, single-digit micrometer or even sub-micrometer spot sizes. And if you compare the typical detection sensitivity, for example, with the generation of ICP-MS instruments that was available maybe 8 to 10 years ago, so basically the past generation of quadrupole ICP-MS, then of course, this was obviously increased already significantly by at least 10 to 20 times when we went to the current generation of single quadrupole ICP-MS. But even on top of that, with the, let's say, introduction of triple quad ICP-MS, basically as an extension to the ICP-MS portfolio, we were able to even further increase the sensitivity for some of the key analytes and also be able to offer superior interference removal at the same time. And the combination of these two, so the general increase of sensitivity over time and the added sensitivity increase using triple quadrupole ICP-MS technology, is really also here helping us to really drive sensitivity and therefore the ability to detect really small quantities of a given element ablated from, for example, a cell or maybe even a thin section of, let's say, an organ by a significant factor. You can see some of the factors roughly estimated here on the bottom of the table. This was probably a very conservative estimation. I think you can potentially even say that depending on the analyte and the sample that you look at, the increases may be even significantly beyond that. Now, also here, a little word about software. Again, this is an important piece in order to control the entire analytical system. And for the elemental analysis products, specifically ICP optical, ICP mass spec, but also multi-collector ICP-MS systems, we do have a common software platform, the Cotegra Intelligent Scientific Data Solution software. And basically this software is also containing a complete feature set to support laser ablation, ICP-MS data acquisition, and also data evaluation. And it works perfectly in conjunction with the HDIP software that was just presented by Cyprian. The Cotegra software is basically a software platform that is allowing you to operate a series of instruments using exactly the same framework with a very user-centric and intuitive architecture. So if you start, for example, with an ICP optical system, you will be immediately familiar with the most important features. If you go to a multi-collector ICP-MS system, although the actual analysis is significantly different. For triple quadruple ICP-MS, which is what I will show mostly for the next couple of slides, the Cotegra ISDF software comes with a specific feature that is the Reaction Finder Method Development Assistant. And this is actually a tool that was developed in order to simplify the process of method creation, especially for users that do not have, let's say, a massive background on ICP-MS. Basically, the method development assistant really does the method development for you. So based off the selection of the element from the periodic table, it really comes with a proposal of the right isotope to measure and also the most appropriate combination, for example, of scan modes and also gas settings, and if needed, also the product ion. If you have experience with ICP-MS and you want to test out different solutions, different approaches, then of course, Reaction Finder also gives you the opportunity to modify these default settings and test all the options that you can imagine. If you want to learn a little bit more about how a triple quad ICP-MS system does work and what the different options are for a variety of elements, there is a TQ ICP-MS toolkit available that is containing the method development guide that you can see here on the screen. Now, let me take you to the laboratory in Bremen. So this is a picture shot in the Bremen facility. What you can see here on the left-hand side is an ICAP TQ triple quadruple ICP-MS system. And on the right-hand side, the Iridia laser ablation system from Teledyne CTech Technologies. We have one of those systems in the lab, and we use it across different instruments and also across a variety of different samples. So for example, the colleagues of the multi-collector ICP-MS do a lot of laser ablation, for example, on geological samples. And on the quadruple base, we do both geological as well as also biological samples. Now, in the next coming slides, I would like to show you a little bit what interference removal and also the sensitivity means for the images that you can obtain with the combined laser ablation ICP-MS system. So this is also some data directly acquired from the laboratory in Bremen. So first of all, this is images from a rat liver thin section. So you can see a microscopic image here on the left-hand side of the slide. And basically in this case, we were interested to compare the performance of a conventional single quad against triple quad ICP-MS specifically for this application. Now, in order to give you a little bit more background on what we are trying to do here, on a triple quad ICP-MS, you normally have much more options. You would be using a variety of different reactive gases in order to, let's say, find the best way of eliminating interferences of your analytes of interest. However, in laser ablation, because we are dealing with very short transient signals, you are most of the time looking at a single mode of analysis because there's no jumping in between. And basically what we did here is we cut the sample in half and then analyzed it using two different modes in order to see whether there are any differences in the performance of the instrument, whether we do the analysis in a conventional single quad ICP-MS mode, or whether we go for triple quad functionality. Now, what you can see here on the image of the left-hand side, basically here, this one on the left is conventional single quad analysis. On the right-hand side, you see the triple quad analysis using oxygen as a reactive gas. The data that you can see here is for sodium, which is an element that you pretty much find everywhere. And here, it really doesn't make a difference whether you go for one mode or the other. Although there's also no particular reason to go for triple quad modes, there are no interferences that need to be eliminated. Nevertheless, the obtained sensitivity is very comparable in both cases. On the image of the right-hand side, we basically look at the same area of the thin section, but we look at iron here. And iron is one of the elements where going for a triple quad ICP-MS does make a huge difference considering the potential sensitivity. In single quad mode, which you can see here again on the left-hand side, we are restricted of going for iron 57 as the isotope of choice here. And iron 57 is one of the minor isotopes of iron. Iron 56 is much more abundant, but it is also very heavily interfered in an ICP-MS. You can see that we can, regardless, eliminate these interferences, but the sensitivity that we see here does equate to only approximately 6,000 counts per second for the area, which is colored here in yellow in the image. If we're looking at the right-hand side, where we do the analysis using triple quad ICP-MS, we can now go for iron 56, so basically leverage a more abundant isotope in nature. We still get, let's say, the same background of, let's say, close to 0 here in this case. We get obviously also the same resolution of the image, but we do see a much higher sensitivity here, basically more than a factor of 10 increased here. And this was all done in a single analysis. One more example that I would like to show you to highlight the benefit of superior interference removal that you can obtain from a triple quad ICP-MS. Basically, you can see here a petiole from a tobacco plant, imaged here for phosphorus, sulfur, and again, calcium. In this case, we were looking again at the difference between single quad ICP-MS and triple quad ICP-MS. But in this case, we operated the single quad ICP-MS also with oxygen as a reactive gas for enabling the full interference removal on phosphorus and sulfur, which are elements that you normally have a high number of difficulties with if you analyze this with a single quad ICP-MS. What you can see here again immediately is that the contrast of the images is significantly better when you go for triple quad ICP-MS, and this is again caused by the superior interference removal of a triple quad ICP-MS over a single quad ICP-MS. You can see this predominantly here for the image for calcium, where basically we do see a significant background coming from other ions created by this sample. And this is directly translated into the signal to noise ratio that has been calculated for some selected regions of interest on these images. So not only is a triple quad ICP-MS able to provide you with a higher sensitivity, it can at the same time also produce a higher contrast in the images. So some other examples, and this is a collaboration with Thea and the London Metallomics facility. In this case, this was a preliminary study to use imaging as a tool to track air pollution. Basically, air pollution is something very typical for our times. A lot of traffic, a lot of small particles that also do potentially contain a number of different metals, and also toxic metals can be part of these. And basically, these metals might be taken up as we, let's say, respirate. And of course, these metals may also cross the blood-brain barrier under certain circumstances. And in this case, the intention of this preliminary study was maybe to look at a sentinel species to find out more about the mechanistic evidence or the mechanistic pathway of how these metals could potentially penetrate into the brain and basically cause degenerative diseases here. So in this case, what you can see here is images of thin sections of eastern gray squirrel brains. These images were created from animals that weren't harmed by any means. They were captured anyway in order to control the population, but they were very useful in order to provide an understanding about how these metals could potentially get into the brains of them and how they could distribute there. And what you can see here is on the left-hand side, you can see an image for phosphorus, which is basically helping us to understand the exact morphology of the sample. And then you can see here iron, copper, and lead images comparing both a location on the outskirts of London as well as downtown London. And you can see in both cases, there is a difference here. You can specifically see that, for example, on the signal of copper. Copper is typically something that could be released, for example, through traffic. And you can see it is significantly more abundant in the image corresponding to the outside of the London metropolitan area. Another example that I wanted to show here today is that imaging is not only able to provide you with qualitative understanding about how certain metals can be distributed spatially across your sample, but under certain conditions, imaging can also deliver quantitative results in case you carry out a calibration upfront. Now, whereas for normal ICP-MS analysis, this is something very straightforward. It is not so straightforward using laser ablation ICP-MS because the response of the sample might be different comparing your standard to your sample. And obviously your calibration is only as good as your calibration standard matches basically the nature and texture and also specifically the ablation behavior of your sample. So what you can see here on the right-hand side, these are now fully quantitative images of Vista rat brains, which were exposed to gadopentate, basically a contrast agent used frequently in MRI imaging diagnostics. And basically, although the gadolinium contrast agents that are used here are, let's say, very, very inreactive, very stable, and normally excreted without further change, you can still see that the gadolinium can still get into the brain and also can create specific diseases. This was done, by the way, with the School of Medicine from Boston University. Now, what the group did was basically develop a strategy to calibrate the system using gadolinium-doped gelatin standards, which were then sectioned to a similar thickness as the sample and behaved relatively similar when looking at the actual ablation process. So basically, when the laser is hitting the surface of the sample, it basically takes away a similar amount of material and also in a similar size distribution. What you can see here, this calibration is actually very reproducible. What you can see here is overlays of 3 independent calibration from 3 different days over a period of 2.5 weeks. And that is actually another very important step in order to make imaging a much more, let's say, widespread and much more widely used technique, because the qualitative information is already very indicative, is already also very important to have, but this can also be complemented by fully quantitative concentrations again. And here you can see it's not intensity that we display in the heat map of the sample. We are actually looking at gadolinium concentrations in microgram per gram. And with this, I would like to thank you for your attention and hand it back over to Thea.
Great. Thank you so much for your talk, and for both of your talks, actually. It was really, really informative. And I hope that kind of opened up the horizons to our attendees to understand a bit about the details behind the techniques that we're using at the LMF. So I think what we will do now is we are going to open up to polling, a little bit of interaction with you guys out there. So I know that we've come up with a series of questions that we would all like the attendees to just answer. I think there's 4 questions. There will be a poll that pops up, hopefully sometime now, soon. And we kindly ask all of you to just answer the questions that are provided there. And that will give us some information to think about. And then I will move on to reading some of the questions that have come in, and feel free also to put in any additional questions that have come up as a result of the talks. All right, so I'll give you a few moments to all do that now. So while those polls are still coming in, just kindly ask to turn all the panelists to turn their cameras on, specifically from Thermo and from Teledyne, so we can have a little bit of interaction for this live Q&A session. Great, so I think we'll probably just wait for like another minute or so for like the polls and the results to come in. And then we have a handful of questions that have come in. And I'm sure that will also spark maybe other questions to enter into the chat as well. Okay, so I think the poll results have come in. It looks, we'll obviously look at the stuff in more detail, but it looks like the elemental analysis is in general interest to most people, that it is performed in-house for a lot of you that have responded, that about half of you seem to have a laser ablation system at the moment. About 38% don't, 11% not interested at all. Okay, totally fine. And the last question, do you plan to invest in this technology within the next year? About 20% said yes, 36% said no, understandable. We all are looking for funding, but 45% of you think that this might be of interest in the future. So thank you so much for those who have joined the poll. And I will go to the Q&A session. So we have a few questions that have come in. So one question that came in actually within the context of Teledyne with Cyprian, your what you were talking about, is the person said, great examples for applications. Just wondering about the method and if it can be applied to any biological samples like plasma or serum samples or anything else from epidemiological studies. So maybe we start with that question. Quite interesting.
Sure, thanks, Tia. That's a pretty good question, and we do have quite a few users in the field who run this kind of analysis. I'm not particularly familiar with the ins and outs of the sample preparation technique, but what they do is for plasma, serum, and also for blood analysis, is the samples are dried basically, and then they're fully ablated with the laser and then quantified that way. So yeah, and I think there are a couple of papers dealing with this application as well, looking at blood, plasma, and so on and so forth. So yeah, in a nutshell, yeah, this is done quite successfully actually, and I can provide a couple of references if needed offline.
Great. The next question that came in is, what are the future prospects of creating an integrated laser ablation ICP-MS system? And I think this is open to everybody here.
I'll take that to start then. I would love to do that. I think what we have currently with two separate systems, as as I think both groups showed from the road mapping of where we got from very low sensitive systems and low speed lasers to where we are today, it's pretty new, that this capability. 2-3 years ago, we couldn't do a lot of what you're seeing already this morning in some of these presentations. So we have the hardware, but we're currently two separate companies, of course, and we have a wide variety of application areas we look into. The software gets us a significant step towards full integration. And by full integration, what I think of is having these tools in the corner of your room, no need to understand what's under the cover of them, just have a GUI, an interface, software interface that allows you to simply and quickly put a sample in there, press a go button and generate some meaningful data. So in that sense, we're on the way down that road, certainly. As to when we can pull together a fully integrated system from one company, honestly, it's simple commercial argument of when the market demands it. So if we find that the bio market evolves at a rate which we expect it to, this, as we say, is a relatively new technology, a new technique, really, then yes, it'll be a very simple argument to put together something like that and move down in that direction.
Any comments from Thermo?
Yeah, maybe to add to this, I think even the status where we are now with the integration of laser ablation with ICP-MS is already a lot improved over where we have been a couple of years ago. So already now we are working with the different manufacturers of laser ablation equipment very closely in order to develop dedicated plugins that really allow to, let's say, flexibly communicate between the two different systems, and also allow for the, let's say, integration of defining the method parameters as good as possible within the workflow. So I think already now, if you want to do the data acquisition, there is already made a good step towards the fact that you have to have only to interact with one system rather than two. And as Damon states, I think to get to a fully integrated system is something that depends on the demands of the markets, and it's also something that we are looking at very closely.
Great. There are no other comments for that question. I'll move on to the next one. So we have another question that came in asking about quantification in laser ablation ICP-MS specifically for biological samples. This is like the $1 million question, I think, that we all discuss and debate. So that's open. What are your thoughts?
That's actually a really good question. And it's seemingly much more difficult for biological samples to be fully quantified using laser ablation ICP-MS compared to geological samples, just because, when you're talking about matrix matching standards, the theory goes that it is easier to find geological samples or standards with a similar ablation behavior. Now, that being said, during the last couple of years, 2 or 3 years, there were huge developments on the quantification side for biological samples using laser ablation ICP-MS. From anything from gels, homogeneous gelatins for imaging experiments, to there's a new technique which will be published relatively recently, and I urge you to keep an eye on that, on those publications coming from University of Vienna, looking at, again, using gelatin as a standard for fully quantitative analysis using laser ablation ICP-MS. So in a nutshell, it is possible. It is much more doable now than it was 2 or 3 years ago. And yeah, I think with the new technologies, it's getting better and better every year to reach full quantification, basically.
There was another question that came in that was kind of on the same lines as this, talking about controls that are used for quantitative laser ablation ICP-MS. So similar to solution work, we use certified reference materials to make sure that the actual numbers we're getting are representative of what's actually in the sample. Can any of you say any thoughts about CRMs within the context of laser ablation?
Well, there are a couple of things. I'll start with a few quick notes and then let the other ones take over. So there are a couple of quick things which are quite important in using standards or gelatin standards or similar standards for biological samples is that they need to behave very similarly. So the matrix needs to be similar in terms of how the ablation behaves. In other words, the volume per pulse, the volume of material that's generated per pulse needs to be roughly the same. And that's quite important. And the other thing is you need to have a really good control over the entire quantity of standard that is transported from the standard itself to the ICP-MS. And with these new techniques, that's actually easy to do. I'm not going to go into the details just because, like I said, there's a couple of papers coming out which detail this quite nicely. But yeah, this is one of the things which is now it's taken care of. So the amount of sample that's generated during the ablation, the way in which the sample itself interacts with the light, and also with the laser beam, and also the wavelengths that's used for this particular sort of analysis. So we don't really need to reinvent the wheels. These are things that were discussed in the past, especially the wavelength for lasers. But the fact that now we can use gelatin standards and similar matrices for fully quantitative analysis is quite important.
I think one additional comment on that, I mean, linking it to the CRMs, having a certified foundation really to base a calibration on. They will come with time. There'll be more and more of them with time. There are a small selection out there already, but following on from what Ciprian was saying, some of these papers which are in press at the moment, and we linked to some of them in the presentation. There's one from Vienna and one from Ghent showing the analytical capabilities. And one thing that's come out of this is that the performance using a glass CRM, a NIST glass, which is the generic laser ablation calibration tool for years, the performance on that NIST glass is comparable to the performance on a gelatin standard. So you can sort of cross-calibrate using the existing CRMs to get a true picture as to what your system is doing from day to day, and then put those CRMs or put those gelatin standards through as QC standards throughout your run. So every however many you wish to run, you can go and ablate a gelatin or a NIST glass as you wish. But the gelatin would be a really good standard to fully certify, I think, as we move forward.
Fantastic. So we have a couple more questions and a little bit more time. So one question that just came in asks, can the high resolution of laser ablation ICP-MS with tissue sections be correlated easily with DESI mass spec imaging analyses of lipid profiles?
Right, so that's actually, nowadays, it's quite easy to do. Especially with HTIP, just because HTIP relies on this sort of multiplexing approach. So you can import images from various sources, and they can be easily correlated. Either automatically, it does have a pretty advanced algorithm for doing that, or the user has full control over how the images are overlaid basically on top of each other. So I think, yes, it is quite easily, especially with the sort of tool that HTIP is right now. And there's a number of other features, and this is one of the issues with not having enough time, because I think this would have been one of the greatest features to present, the fact that, or how HTIP deals with images generated via a multitude of techniques, and how they can work inside HTIP to get relevant data out of multiple methods, and then correlate that with laser ablation ICP-MS data.
Any thoughts from Thermo side?
Not specifically on DESI. I think the comment that Ciprian made here is I would perfectly agree to that.
Okay.
I think, Theo, as well, I think with that, we can expand from that. I mean, DESI is one example, and Ciprian mentioned that there are other image technologies you can bring and import in and look at how we can integrate all of these images into something meaningful. But the other side of this is linked to how you can use the ICP technology as well, in that, as Daniel showed, there's a variety of ICP technologies available, multi-collectors, single collectors, triple quads, and so on. And there's already papers published, I think, by the Thermo group who we have here as well, showing how you can split stream. So you can have one sample in a laser being ablated, sending that off to different ICP technologies to get a whole range of sample data from the multi-collectors for the isotope ratio, high precision isotope ratio work, if that's of interest, through to optical. In fact, we can actually do this with optical technologies as well, get highly well calibrated data, high quality data out of optical. So there is a range of techniques we can use from this, both with software and with hardware, to bring all this data together.
Great. So yeah, the questions are still coming in. So the next question is LMF already integrating these technologies? Yes, we are pushing for all this type of integration. So we do have this DESI system that I mentioned in my talk. And we are very keen to see how we can use HJP to start integrating these different modalities, and also working with people at King's to develop machine learning and algorithms to integrate those data sets as well. So yes, this is in the pipeline. This is the whole point, I think, going forwards. The next question is, forms of quantification discussed here now have been using external calibration. Have any other strategies been examined, like standard addition or internal calibration?
How much time do we have? Because this is a really interesting question. It just requires a lot of time to properly answer.
We have probably about like 3 more minutes until give people about 10-minute break for the afternoon session, so we can spill it.
Okay.
3 to 5 minutes.
So yeah, so there are a lot of, well, there's a few techniques that rely on standard addition and internal calibration, or internal standard calibration. I'm particularly fond of one technique which was developed, I think, I can't remember the university. So they were printing a standard on top of the glass slide, and then mounting their sample, and then fully ablating the sample and the standard underneath without ablating the substrate. And because it was full consumption, the aerosol was mixing fairly well, and you would have that as an internal standard. So that works pretty well, and that was published a couple of years ago, and it's quite a highly cited paper. Other forms of calibration, you can certainly add solution and desolvate it. So basically, make your standard solution, run it through the desolvator, and mix that with the aerosol that comes from the laser. It's a bit more, it's not as straightforward as it sounds, but it does work pretty well. And again, it's not something that we've invented yesterday. It's been around for a couple of years, and it's fairly well-established technique for calibrating.
Great.
I would like to make an addition here. There's another calibration strategy, which is maybe not so commonly known, which could also be really helpful here in this context, and especially since you mentioned the addition of an aerosol to the ablated material. Mm-hmm. That obviously combined with the isotopic capability of ICP-MS, basically the ability to analyze different isotopes of one and the same element, allows the application of isotope dilution for quantification purposes. And this is actually a very attractive solution, because the actual result of the quantification only depends on the measured isotope ratio, and not so much anymore on external factors. And it's typically a calibration strategy that can be well applied to analytical protocols that do require, for example, an extensive sample preparation, or that are also prone, for example, to drifts across the measurement. This is already something where isotope dilution can provide a significantly increased accuracy in the determination.
Great, perfect. So that was the last question, and we are smack dab on time. So I just want to give both Thermo and Teledyne kind of an opportunity to say some closing remarks, however you want to address the attendees before we take a 10-minute break and jump into our afternoon session. So maybe we'll start with the same order. Damon, you can say something, and then we'll end with Daniel.
No, as I said during my brief introduction, I'm learning all the time. Bio is quite a learning curve, largely because of the big words you all use, which is fascinating, but I do need a dictionary with me sometimes. But it's incredibly important for me to understand not just what you can do with it, but what you can't do with these tools. So as you're looking through the rest of these presentations this afternoon or in the future, I believe they're recorded and they'll be available online afterwards.
Some of them will be, yeah.
If you have any questions, however stupid you think they are, ask us. You have our contact details in here, so please contact us and ask us. I'd love to have a discussion about not just what you can do, but what you can't yet do, and see if we can, together with our partners like Thermo, we can find a way to help resolve some of those issues.
So I would like to first of all thank you, Thea, for the opportunity to be here to give this presentation to you. I would also like to thank everybody who stayed on during the lunch break. I do appreciate that this is quite a long day, and thank you for spending the time with us. And I can only echo what Damon mentioned. We are here to learn more about your applications, learn more about your science, and we are definitely here also to be in contact with you to answer your questions. So if there is anything that isn't fully clear, anything that you would like to know, just give us a shout, ping us, and we'll be happy to answer.
Great, thank you guys so much. You can find all contact details in the Symposium booklet, which we've been submitting links to in the chat all day long. So thank you all for your attention. We're going to have a short break now, and then our afternoon session will be starting up. And we will see you in about 8 minutes or so. Thanks so much.